So this is chapter 3, which is another chemistry chapter in unit 1. We're going to talk about organic molecules, functional groups, polymers and monomers, how to make polymers and monomers, and macromolecules important to biology. So organic molecules, that means that the atoms are held together by covalent bonds. So the atoms in organic molecules are sharing valence shell electrons. And also organic molecules contain carbon and hydrogen. So we might say they have a hydrocarbon backbone. Things like sugars, fats, and proteins would be organic molecules. Also, something like methane, which is CH4, is an organic molecule. Don't get confused with the meaning of organic in chemistry and the USDA organic as it refers to your fruits and vegetables. Like an organically grown strawberry and a conventionally grown strawberry both contain organic molecules in them. Inorganic molecules generally don't contain carbon and hydrogen. They could have either ionic or covalent bonds. So carbon dioxide is CO2. That has carbon, that has covalent bonds, but no hydrogen, so not organic. Water is H2O. Water has hydrogen and covalent bonds, but no carbon, so water is inorganic. Something like table salt is sodium chloride. That doesn't have covalent bonds, it's ionic, and it doesn't have carbon, doesn't have hydrogen, so table salt would definitely be inorganic. So that brings us to monomers and polymers. Many organic compounds are polymers. What does that root word poly mean? That means many. So a polymer is a chain of monomers. What's the root word mono mean? It means one, right? So this is a cheesy example, but this paperclip chain here is a polymer, and each individual paperclip that makes up the chain would be a monomer. Typically, polymers are synthesized by dehydration reactions, which are also called dehydration synthesis or condensation reactions, or broken down by hydrolysis reactions. So dehydration synthesis is named dehydration synthesis because you remove a water, an OH from one monomer or one building block, and an H from the other. So OH plus H is H2O. So you remove a water and link those two monomers together. So that's a dehydration, right? Removal of water. Synthesis, because you're combining things. So a real life example of that would be if we take glucose and fructose and enzymes that remove the OH and H, so remove the water, we get sucrose. The opposite of dehydration synthesis is hydrolysis. The root word hydro means water, and lysis means breaking apart. So with the help of enzymes, you add an OH to one side and an H to another, and you split the polymer into two monomers. So that would be taking the sucrose, enzymatically adding the water in, the OH on one side, H on the other, and getting two monomers, glucose and fructose. So when you get to organic chemistry, you'll probably talk about functional groups. Functional groups are worth kind of learning about because they're groupings of atoms that occur commonly in many organic molecules. And if we know how functional groups respond or act, that gives us an idea of the overall properties of that particular molecule. So here's a few functional groups that you're going to run into again and again in biology. The first one is a carboxylic acid group. It's a COOH group. So these are important in amino acids and fatty acids. They can also release hydrogen ion and act as an acid. Then there's amino groups. Amino groups are this NH2 group. These are important because they can either accept or release hydrogen ions, and they can form bonds with other molecules. We're going to see amino groups in amino acids. There's also that OH group that we've talked about before, the hydroxyl group. This is important for dehydration synthesis. It's important for hydrogen bonding. Also, hydroxyl groups affect an overall molecule solubility in water. You're going to find hydroxyl groups in things like carbohydrates, fatty acids, and amino acids. Then we've got this PO4 group. That PO4 group is a phosphate group. You've maybe heard of ATP, or adenosine triphosphate. That has 3 phosphate groups in it. Phosphate groups are important in higher energy compounds. They're also important for phospholipids, which make up our cell membrane, and nucleic acids, which would include DNA and RNA. So these are just some functional groups that are important in biology. So let's review. What does the word organic mean in biology and chemistry? So what kind of atoms are in organic molecules? That would be carbon and hydrogen, right? Also, what kind of bonds do they have? That would be covalent bonds. Ooh, this is hard to write. Covalent bonds. On a test or something, please write in full sentences, but you wouldn't be writing poorly with your mouse. So what's the term for the reaction that breaks polymers into monomers? So what's a polymer again? The polymer is made up of many monomers. So if each of these blocks is a monomer, right? The polymer is many monomers, and we're looking for the thing that breaks that down. So to break down the polymer, we have to use a hydrolysis reaction. Hydro means water. What's lysis mean? Lysis means breaking apart. So hydrolysis reaction breaks polymers into their monomers. So let's go over some biologically important macromolecules. The first macromolecule we're going to talk about is the carbohydrate. A lot of these macromolecules, like if you've ever looked at a food label, you've probably seen some macromolecules before. So carbohydrates contain carbon, hydrogen, and oxygen, and they're called carbohydrates because there's a 1 to 2 to 1 ratio of carbon to hydrogen to oxygen. So they're CH2Os, or carbohydrates. Somebody thought they were really clever when they came up with that name, right? So broadly, carbohydrates are important because they are great sources for chemical energy, and they can also be used for structural support. There's 3 classes of carbohydrates. There's monosaccharides, disaccharides, and polysaccharides. Monosaccharides, the root word mono means single, right? Saccharide means sugar, or saccharon means sugar. So these are simple sugars. These are the monomer of a carbohydrate. For example, glucose is a monosaccharide, and they can either be in this straight chain form or the ring form. So then we've got disaccharides. That root word di means 2. So that's 2 monosaccharides linked together. What reaction would build a disaccharide? That would be a dehydration synthesis reaction. And what reaction would break down a disaccharide? That would be hydrolysis reaction. So sucrose is a disaccharide made up of the monosaccharide glucose and fructose. Maltose is a disaccharide made up of 2 glucose monosaccharides. And lactose is made up of galactose and glucose. Don't memorize those, but do know that a disaccharide is made up of 2 monosaccharides. Polysaccharides then, that root word poly means many. So polysaccharides are long chains of many monosaccharides and disaccharides all connected together. You've maybe heard of starch, like a starchy vegetable before, like a potato. So that's how plants store their carbohydrates, and glycogen is kind of the animal equivalent of starch. That's one of the ways that animals store their carbohydrates. So let's review. Which of these would you expect to be larger, a polysaccharide or a monosaccharide? What's the root word poly mean? Many, right? So the root word mono means 1. So a polysaccharide is going to be bigger because it's made up of many monosaccharides. What are two functions of carbohydrates? So structure, like plant cell walls, are made up of carbohydrates. Also, energy storage, short-term energy storage would be another carbohydrate function. So we did carbohydrates. That brings us to lipids. Lipids also contain carbon, hydrogen, and oxygen, but they're not in that cool 1 to 2 to 1 ratio that carbohydrates are in. Also, lipids are kind of a large, loosely defined group, so their underlying structure really varies. Like, there's not one specific monomer that makes up lipids, but lipids all have little or no affinity to water. Remember before we talked about oil being hydrophobic. So lipids are important for energy storage. They're a major component of all of our cell membranes. Some of them can act as chemical messengers, like if you've heard of a steroid before, that's a chemical messenger. Some example lipids include fatty acids, phospholipids, and glycerides. And technically, some lipids are too small to be considered macromolecules, but don't get too hung up about that. Just if you're reading the book and the book says lipids are macromolecules, like, some lipids are too small to be considered macromolecules, so that's technically correct. But if we think about like macros and nutrition and things like that, lipids definitely fall into that category. So an example lipid would be a fatty acid. Fatty acids have a long hydrocarbon chain. The head has a carboxylic acid group, which is that COOH group. The head region is hydrophilic, so the head region readily interacts with water, and this tail region does not readily interact with water. It's hydrophobic. There are saturated and unsaturated fatty acids. So saturated fatty acids are said to be saturated because they contain single bonds between all of the carbons, so that they have the maximum number of hydrogens that these molecules can fit. So their hydrocarbon tails are straight, so they stack up nicely and they are solid at room temperature. So animal fats tend to be saturated fatty acids. Unsaturated fatty acids, on the other hand, have double bonds between some of the between some of the carbons in these hydrocarbon tails. And so these hydrocarbon tails are bent and don't stack up nicely. So these are going to be liquid at room temperature. So plant oils tend to be unsaturated fatty acids. That brings us to another kind of lipid, which is the glyceride. Glycerides are made up of fatty acids that are attached to a glycerol. So just like everything else we've talked about, we synthesize these with dehydration synthesis and break them down with hydrolysis reactions. Glycerides are important for energy storage, insulation, and protection. So generally when you're thinking about fat, you're probably thinking about a triglyceride, which would be a glycerol and 3 fatty acids. There's also diglycerides. What's the root word di mean? 2. So that's a glycerol and 2 fatty acids. And a monoglyceride would be a glycerol and 1 fatty acid. Another kind of lipid is a steroid. Steroids have these kind of 4 carbon rings, and then there's different functional groups that can be attached to those rings. So cholesterol is a steroid, and cholesterol really gets a bad rap for clogging arteries, but cholesterol is important for stabilizing our cell membrane. It's also important for cell growth and division. Hormones like estrogen and testosterone are also steroids. Those are important for cell signaling and regulating metabolic and sexual functions. So then we've got the phospholipid, which is another type of lipid. It has a hydrophobic portion and a hydrophilic portion, and it's very important in the cell membrane. So this head region here, this head region is hydrophilic, and these hydrocarbon tails are hydrophobic. So phospholipids can actually form bilayers, which is 2 layers of phospholipid, or micelles in water. So we'll talk more about the cell membrane in, I think it's chapter 4, but phospholipids are an important component of the cell membrane. We have a phospholipid bilayer making up a good chunk of our cell membrane. So what are some functions of lipids? So I think when people think lipids, they think fats, right? So insulation, protection, longer term energy storage, those would all be example functions of lipids. People tend to forget cell membrane structure and chemical messenger. So those are several functions of lipids. What type of lipid is responsible for separating the inside of the cell from the outside of the cell? That's going to be a phospholipid. Phospholipid. I don't know if that's helpful to read, but phospholipid. And would you expect a lipid to be soluble in water? No. Maybe portions of it are, but it is not entirely soluble in water. So we did carbohydrates, we did lipids, now we're on to proteins. Proteins contain carbon, hydrogen, oxygen, nitrogen, sometimes phosphorus, sometimes sulfur, just kind of depends. The monomer, or the building block of a protein, is an amino acid, and the specific bond between amino acids is called a peptide bond. Technically it's a covalent bond, but it's a special type of covalent bond called a peptide bond. Proteins have all kinds of functions. We're going to go over several on the next couple slides. And they have a really critically important, like, 3-dimensional shape, and that shape is important to the protein's function. So we'll talk more about enzymes in, I think, chapter 5 in unit 2, but enzymes help increase the rate of chemical reactions, and many enzymes, like digestive enzymes in our body, are proteins. Proteins can also store energy, so casein is the protein of milk, ovalbumin is the protein in egg whites, so there are storage proteins. There's defensive proteins as well. I think you've probably already heard of antibodies before. So antibodies are part of our immune system, and they help inactivate and destroy viruses and bacteria. There's also proteins that are important for transport. I think many people have heard of the protein hemoglobin before in our red blood cells that helps transport iron around. There's also proteins in our cell membrane that move material in or out of the cells as well. Insulin is a hormonal protein, so some hormones are steroids, which are lipids, and some hormones are protein-based. You can also have receptor proteins, so one cell can communicate to another, like a nerve can talk to a muscle with a neurotransmitter, and then that neurotransmitter binds to a receptor protein. There's also proteins for movement. In your muscles, there's proteins called actin and myosin that are really responsible for the contraction of the muscle. And then there's other structural proteins like keratin in your hair and nails, and collagen in your skin. Those are proteins as well. So proteins are made up of amino acids. Amino acids have 3 parts. They have the NH2 group, which is the amine group. They have the COOH group, which is the carboxylic group. And then they have an R group, which is really kind of the rest of the molecule. The R group can either be hydrophobic or hydrophilic. It really just depends on the particular amino acid. So with protein structure, there's primary, secondary, tertiary, and quaternary structure. Primary structure is the first level of structure. It is the order the amino acids are in the protein. So each of these colored spheres is supposed to represent a different amino acid in this protein. So if they were in a different order, there would be a different primary structure for this protein. The secondary structure happens when the amino acids start to hydrogen bond with each other, and it forms these pleated sheets or alpha helices, which I guess my pleated sheet sort of looked more like the alpha helix, but you get the idea there. They're hydrogen bonding with each other. The next level is the tertiary structure, and this is when the amino acids start to interact with the surrounding water molecules. So the amino acids are hydrogen bonding with the water around it, and you get this kind of compact, kind of globular shape. And then with quaternary structure, this is when you have more than one amino acid chain. So in this picture, you can see kind of the light blue chain and the dark blue chain are interacting with each other and hydrogen bonding with each other. It's really common for proteins to have multiple amino acid chains. For example, hemoglobin in your red blood cells is made up of 4 chains of amino acid. So speaking of hemoglobin, even just like a simple change in one amino acid in a protein can affect how those hydrogen bonds between the amino acids occur, and so that can affect the shape of the molecule, which can affect the function of the molecule. So a normal hemoglobin again has those 4 subunits. There's 2 beta subunits and 2 alpha subunits, and they carry oxygen around, and those proteins don't clump together, and so you get this normal red blood cell. They call the shape of a red blood cell a biconcave disc. You can only see like one part that's concave here in the picture, but the other side has that same kind of divot as well. In sickled cells, there's one amino acid different. So in the secondary and tertiary structures, that protein or that subunit of the protein kind of folds up differently, and so that causes the hemoglobin protein with all 4 subunits to fold up differently, and those beta subunits are able to aggregate with each other, which actually you don't really want, and so those proteins aggregating together causes the red blood cells to have this abnormal sickled shape there, and that can cause problems with transport of those red blood cells through the capillaries, which are those smallest diameter blood vessels. So proteins can also be denatured. When a protein's denatured, that means some of the hydrogen bonds are broken apart, so the protein's shape and function deteriorate. So down here in the bottom, there's like a normal twisted up protein here, and then it's exposed to some sort of extreme condition, like extreme of temperature or extreme of pH, and that's a denatured protein. Also, in this frying pan up here, these eggs, right? The ovalbumin in the eggs is denatured. So, you know, if you're making a cake, right, like you would not put these eggs from the frying pan into your cake mix because it's not the same. You're going to put the liquid eggs into your cake mix, and then when you bake your cake, the proteins will denature that way too, but it'll denature in the way you want it to denature in a cake, rather than having like clumpy fried egg chunks in your cake mix. So denaturation can be problematic because when the protein's structure deteriorates, its function deteriorates, and so this can be damaging to tissues and organs. So review number 8 says, why is the structure of a protein important? That would be because structure and function are linked, right? Number 9, what type of chemical bonding is responsible for the primary structure of proteins? So what's the primary structure? That's the order of amino acids. So what holds amino acids together in a protein? That's a peptide bond, which is technically a type of covalent bond, but I was looking for peptide bond. What kind of bonding is responsible for secondary structure? That would be a hydrogen bond. Remember, secondary structures when the amino acids start to fold with each other, so that's hydrogen bonding that does that. And then number 11, what is the monomer that proteins are made out of? That's an amino acid. Amino acid is the monomer of a protein. Here's a silly joke, super cheesy, that's like, why is that protein so grumpy? It's made up of amino acids. I don't know if that will help you remember or if that's just humiliating for me. You decide. So that brings us to nucleic acids. Nucleic acids are an organic molecule, so they have carbon and hydrogen. They also have nitrogen, oxygen, and phosphorus. The monomer of a nucleic acid is a nucleotide, and specifically there's 5 kinds of nucleotides. There's adenine, guanine, cytosine, and thymine in DNA, and then RNA has adenine, guanine, and cytosine still, but it has uracil instead of thymine. So nucleic acids are important for storage and transfer of information in cells. If you've heard of DNA and RNA before, literally the NA in their name stands for nucleic acid. So DNA is deoxyribonucleic acid and RNA is ribonucleic acid. So if we look at the monomer of the nucleic acid, the nucleotide has a nitrogenous base, and that determines which nucleotide we're dealing with. It also has a pentose sugar, which is a 5-carbon sugar, and a phosphate group. And adjacent nucleotides are joined by something called a phosphodiester linkage. So in DNA and RNA, the phosphates and sugars are hooked together, and we do that with dehydration synthesis, and we get a sugar phosphate backbone of the nucleic acid, and then the sequence of information is actually carried out in these nitrogenous bases here. So you've probably heard of DNA before. Well, and if you hadn't, we talked about it in chapter 1, so you have heard of DNA before. DNA is 2 chains of nucleotides, and they twist around each other and form what's called a double helix. These chains are anti-parallel, meaning the 5' end of one runs counter to the other one. So like in this picture, do you see how this phosphate group is up here on this chain, right? So the top, and on the other chain, it's at the bottom. That's what we mean by anti-parallel. They also have complementary base pairs. So adenine pairs with thymine and cytosine pairs with guanine. They pair together because of the number of hydrogen bonds. There's 2 hydrogen bonds, see 1, 2 between adenine and thymine, and 3 hydrogen bonds between cytosine and guanine. And one DNA molecule can actually hold many genes, and broadly speaking, genes code for a particular polypeptide sequence, and really that's basically saying like genes code for a particular protein sequence. RNA, on the other hand, is usually a single chain of nucleotides. So we'll talk more about this in Unit 3, but to kind of get you thinking about some big picture information, right? Your DNA here contains information that codes for the synthesis of mRNA, and then that mRNA goes to an organelle called the ribosome and directs the synthesis of a polypeptide or a protein. So when we go from DNA to RNA to protein, we are expressing the gene in DNA. And again, we're going to talk more about protein synthesis in Unit 3 and gene expression in Unit 4. So don't worry if that doesn't all make sense just yet. So let's compare and contrast DNA and RNA. So molecule type, both DNA and RNA are nucleic acids, so they're both nucleic acids. Oh man. How many strands in DNA? There's 2 strands in DNA and 1 strand in RNA. Nucleotides in DNA, which one is only in DNA? So thymine is only in DNA, and which one is only in RNA? Uracil is only in RNA, but they both have adenine, cytosine, and guanine. And then don't worry about the sugar. I got overexcited in this. I thought we had talked about it, but we didn't. So I won't test you on the sugar. But if you're curious, the sugar in DNA is deoxyribose, and the sugar in RNA is ribose. So we've kind of talked a little bit about genomics and proteomics already, and we're going to talk about them more in Unit 4, but there are fields of study related to genes and proteins and whole gene sets and whole protein sets. So genomics analyzes the genome, which is large sets of genes, and DNA sequencing, there's actually several different ways we can do DNA sequencing, several techniques, but that's going to determine the sequence of nucleotides or the order of nucleotides in DNA. Proteomics then looks at protein sets and compares the proteins expressed in different cells or tissues or organisms. So we could look at the proteins expressed in a cancerous cell versus a non-cancerous cell. And then with all of this information from genomics and proteomics, we've got bioinformatics, which uses computer software and other analytical tools to store and analyze all of this data. Let's review. Match the biochemical compound to the picture and provide an example function. And I've got the biochemical compounds or macromolecules listed for you there, the carbohydrate, lipid, protein, nucleic acid. So maybe take a pause here and try to do it before you just listen to the answer. So if I were a student taking a test, I would look at these and try to start with whichever one I thought was easiest first. So are there any of these that maybe seem more recognizable to you? I feel like for most students it's going to be this double helix here in D. So D is going to be our nucleic acid. So we've got the biochemical compound, and then we needed to have an example function. So DNA and RNA store and transfer information. So I'm going to write store info, which again, if you were on a test, you would need to give me a little bit more than, store info, like say store and transfer information, something like that. I'm just writing it very shortly because it's very challenging for me to write with the mouse. Okay, so we got nucleic acid done. Is there another one that seems easy to you? One kind of tricky thing with this question is, like, in biology, you don't have to have a chemistry prereq for this, and none of these molecules are presented to you in the same way. I think another relatively easy one, or, the second next one would be the carbohydrate, which is B here. So carbohydrates B, our nucleic acid was D. So you might be like, well, I tried to count the carbon and hydrogens to get the 1 to 2 to 1 ratio, but in this kind of drawing, everywhere the lines intersect, there's a carbon assumed there. So that makes that a little trickier to count to your 1 to 2 to 1 ratio. Of carbon, hydrogen, and oxygen. But this is a carbohydrate. Carbohydrates are good for structure, and they're good for energy storage, like more short-term energy storage. So then this next one, A is our protein. It's made up of multiple amino acids, and it has a NH2 group on one end and a CO, it should be OH group on the other end, right? But they got this extra H from this side over there. But that is our protein, and proteins have all kinds of functions, right? Proteins, antibodies are proteins, so there's defensive proteins, there's transport proteins, there's structural proteins. Proteins important for movement, like they run the flagellum. They are important for contraction in muscles, things like that. And then C is our last one, and that's going to be the lipid. So the lipid is C here. This lipid in particular is supposed to represent a phospholipid, but they did it a little bit poorly because it's not just a big phosphorus in the head group here. It's even more than just a phosphate group, but that's an example of a lipid. What are some lipid functions? Lipids do more long-term energy storage. They do insulation, protection. They also are important, like the phospholipid would be important for cell membrane structure, and then steroids are lipids as well, and those can be chemical messengers. So those are the various functions of lipids there. So we made it to the end of chapter 3. We talked about organic molecules. Remember, to be an organic molecule, the molecule needs to have carbon and hydrogen and covalent bonds. We talked about the different functional groups. Don't get super hung up in memorizing those for the test, but it is worth kind of knowing some of the functional groups because you'll see them again and again, right? Phosphate groups, you're going to run into that again. Amino group, carboxylic acid, hydroxyl group, those kinds of things. You're going to see them multiple times. Polymers and monomers, what's the root word poly mean? Many, right? Mono, singular. So a polymer is made up of many monomers. So for example, a polysaccharide is made up of many monosaccharides. Dehydration synthesis reactions take a water out and link monomers together to build a polymer. A hydrolysis reaction is just the opposite of that. That's where you add a water in, hydro, and lysis, split that polymer apart. We just went over carbohydrates, lipids, proteins, and nucleic acids on the previous slide. Make sure you know those. Make sure you know the different kinds of nucleic acids and the different kinds of lipids and the different kinds of carbohydrates. Make sure you know the monomers of those if it's applicable. So like carbohydrate, monosaccharide, protein, amino acid, nucleic acid, nucleotide. We talked a little bit about genes and gene expression, but don't get super hung up on that just yet. A gene is a segment of DNA that, we'll say for now, codes for a protein, and when that gene is expressed, that protein is produced. That's the short story for now. We'll talk more about that coming up later. We also very briefly talked about genomics, the study of genomes, which is sets of genes, and proteomics, which is the study of sets of proteins or proteomes, and then DNA sequencing tells you the order of nucleotides in DNA. So that's it for chapter 3.